Okay, it's going. All right, here we go. Friday afternoon on a rainy day. Let's get this done, then let's get the heck out of here. Okay, we're discussing recombinant protein expression. So I picked a paper here on recombinant protein expression of Cas9, so it connects to also our CRISPR lectures. So this is in the Journal of Biotechnology on the expression of recombinant Cas9 protein in E. coli, BL21DE3, and BL21DE3 Rosetta strains. This paper is not what I would call like a high impact paper. This is more like a methodology, kind of like nerdy paper, but it's easy to understand. And I think it's a very useful paper in that it outlines a lot of the problems that you have to optimize when you're dealing with recombinant protein expression. So I thought it was actually a decent paper to go through and read. Okay, so I pulled out some highlights from the paper. I figured we'll just go through each of these and do a little bit of teaching in between. Okay, let me read this first thing. The system is composed of Cas9. Okay, so this is where I'm supposed to review. So just for review, remember there is a protein called Cas9, which is endonuclease. That's one part of the CRISPR system. The second component of the CRISPR system is the guide RNA that targets the Cas9 endonuclease to cut, and it cuts DNA right down in the middle of the molecule, which means it's an endonuclease, and it creates what are called double-stranded breaks. Okay, so that should be review. We all remember that. Okay, so more interesting things about the system. The Cas9 protein is 158 kilodaltons. So you guys have probably not run enough STS page gels to sort of just readily, quickly interpret this. But when you run an STS page gel and you run a ladder, usually the largest ladder is 250 kilodaltons, something like that. Second one, maybe 200 kilodaltons. So 158 kilodaltons, there's usually about 150 ladder right at the top. So my first question is this a large protein or a small protein? It's a very large protein. Yeah, so maybe not the largest, but sort of like in the cell, there's always like extremes, but kind of like the largest stuff is like greater than 200, but that's pretty rare to be that large. So 158 kilodalton protein is actually pretty big. And so let me ask another question, which is if you're going to have to express recombinant proteins and make high yield proteins, do you think it's probably easier or harder to express a large protein? Yeah, it's more difficult because it's harder to get high yields of heavy, large proteins. So that's something to just quickly understand about the system. But it's a very important protein that needs to be optimized and expressed for many different reasons. So it's important to study this. Let's see. Here we systematically, okay, so this is sort of like telling you pretty much the gist of the paper. Here we systematically evaluated the expression of recombinant Cas9 protein in 2 different E. coli strains in complex and defined media. So we'll go through that. Quickly, let's make a quick list. What do you guys think is the purpose of the paper? Yes, so you said see which of the 2 strains produces the highest yield. And then what was the other one? Somebody else was going to say something? Test media, test different media. Let me see what I put. Optimize the expression of Cas9, test different E. coli strains and media. So you guys said exactly what I said. So we're all on the same page. That essentially the gist of the paper is these 2 points. Okay, let's also just finish. Will somebody open the door for him? Let's also just answer this question. Why would you want to make recombinant Cas9? For what purpose? I can't quite hear you. Well, use it how? Use it for what precisely? So that's an interesting thing. So so again, like this is helpful for me to clarify. So you said 2 things. You said in vitro versus in vivo. So in vivo usage, that means like in the living organism. In vivo usage of Cas9 would probably be something like a transgenic insert of Cas9 into the genome, although you can use recombinant for in vivo. In what case are you using recombinant Cas9 in vivo? So this would not be recombinant protein expression that you purified from E. coli. This would be sort of like transgenic insertion. What's the usage case where you're making recombinant Cas9 from E. coli, purifying it, and then using it in vivo? What does that actual usage case look like? Yeah, for what purpose? Yes, microinjections. Okay, that's essentially like why you would do this. So why would we do this? Big reveal, microinjections. Okay, so this is going to be used for if you're harvesting embryos of, say, zebrafish, or harvesting embryos of cows, or harvesting embryos of Drosophila. Anything you're going to do microinjections to do genome editing, this would be a useful, it would be useful to be able to have highly purified, concentrated frozen stocks of Cas9 that you could just pull out of the freezer, thaw out, and microinject. Okay, so that's what the purpose of this, what you would use it for. Let's see, what's this? The recombinant protein showed, okay, so this is just their results. Okay, so the two cell lines that they are comparing, one of these is called BL21DE3, and another one is called Rosetta DE3. And in the last lecture, I went into a pretty good detail about how different strains is very important for getting good expression of different proteins, okay? So I looked up what the genotypes of these strains were. So anybody can do this, and now you should have the confidence that if you saw a paper talking about a strain, you can just Google that strain and you'll find websites that will tell you everything you need to know about that strain. So I Googled BL21DE3. Here is the genotype. So here you see it tell you it's E. coli, and then these individual things, these are the things that make it unique. Every organism has its own like mutation nomenclature, okay? But if it's listed in the genotype, typically these are deletions. So this is a deletion of that OMP-T is a deletion of OMP-T. So, but they're not annotating these with the actual deletion symbol. Sometimes these, what would you call them, like codes? Sometimes they reference a gene by name. Other times they need to be looked up. So if you were really, really intent on really learning exactly what these genotypic annotations were, I would actually take the actual text, like whatever this thing is, HSDS, RB minus, RB, MB minus. I have no idea what that means, but you could probably take that text, that string, and you could search that, and you could probably find out somewhere exactly what that is. The only thing I would suggest is that these are definitely gene names. And probably these are different deletions, and this is probably a very specific deletion of whatever HSDS is, something like that. Just understand that you can look up the genotypes, you can find out what they do. And then they have often annotated notes on the genome that tells you like a basic gist of like what it came from, what it does, why it was designed. So it says the strain contains DE3. DE3 is a lambda prophage. I think we did talk about lambda. Remember, lambda is a phage that can integrate into the genome, carrying the T7 RNA polymerase gene. So when you see DE3, it's saying that DE3, that nomenclature means that it has an insertion of a lambda phage, which includes a gene called T7 RNA polymerase, okay? And this will become important later. And this gene is under the control of a LAC UV5 promoter. So there's some promoter, which is essentially a variant of the LAC operator. Okay, so we know that it's controlled by IPTG induction somehow, although it's a little bit more complicated. And so it says exactly that. It says IPTG is required to induce expression of the T7 RNA polymerase. Okay, and also, let's continue. The strain does not contain the LAN protease, which I taught you, and it's also deficient in the outer membrane protein protease OMP-T. So it's got two proteases that are deleted. That's the derivation from the BL21. Okay, so that's pretty understandable. I'm just pointing out here that when you see these like coded language things in the text, it's really quite easy for you to take that and Google it and figure out exactly what it is, and you should have the knowledge that you could be able to interpret that. Okay, the next strain, which was Rosetta DE3. So this is essentially just going to be the BL21 DE3 plus the P rare plasmid that we discussed earlier. So if you look at the actual notes right here, it talks about it. Host strains are BL21 derivatives enhanced for expression of eukaryotic proteins that contain codons rarely used in E. coli. These strains supply tRNAs. So remember when I described that plasmid, it shows you here, these are the actual tRNAs that are encoded in that plasmid: AGG, AGA, AUA, CUA, CCC, GGA. So that's 1, 2, 3, 4, 5, 6. So that's actually 6 codons. When I presented it last time, I think I said 10 to 20, so that was incorrect. Here we're actually fact checking it. The precise number is 6 tRNAs. Maybe codons isn't correct the term. 6 tRNAs that are actually provided by this plasmid. Thus, Rosetta strains provide for universal translation, which is otherwise limited by rare codons in E. coli. Okay, so that's the gist. And here they're telling you again about the DE3. DE3 indicates that the host is lysogen, not quite. I think that means that it has incorporation of the lambdaphage DE3. And there it carries a chromosomal copy of T7 RNA polymerase. So here's what you should kind of know now is that if you see DE3, that means it has T7 RNA polymerase integration. So now let's actually talk about that. We'll do a little bit more teaching. This is pertinent to PET plasmids. So then what you're seeing with DE3 is actually telling you that they are compatible with PET plasmids. So I'll just quickly define and draw out the PET plasmids. Go over here. Okay, so let's do a little annotation of the PET system. The PET system is regulated by the T7 promoter. Okay, so in a PET plasmid, you have a multi-cloning site where you insert your gene X. PET plasmids are almost universally for recombinant protein expression, so they're optimized for this specific purpose. And they actually have 2 regulated promoters, not 1, but 2. One of these is called the T7 promoter. That's a more outside promoter. And the internal promoter is that special lac operator. Okay, so there's essentially a variant of the lac operator, 3', well, I guess not 3' of the gene, but closest to the gene start codon. And then upstream, 5', there's a second promoter, which is a T7 promoter. Okay, these plasmids, so this is the unique sort of setup that sort of defines the PET system. The PET system is uniquely compatible with these DE3 lines, which require an insertion of the T7 RNA polymerase. So in strains of E. coli that are compatible with PET plasmids, they have a genome, an internal genome that has somewhere an insertion of the T7 RNA polymerase gene. Okay, and if you read the details of these insertions, usually these insertions are regulated by a promoter, which is also a variant of the lac operator. Okay, so we have in this system, there's 2 instances of promoters that are the lac operator. Okay, so let's go through what actually happens, and you'll see the unique features of this system. So first, let's talk about this T7 RNA polymerase gene. So that's going to create a protein, which is a T7 RNA pol. And we should all know, so what's the function of a T7 RNA polymerase? Or maybe more broadly, what's the function of an RNA polymerase? Yeah, so this does transcription, okay? So it's a protein that's doing transcription. So it's a DNA-dependent RNA polymerase doing transcription. The T7, the fact that it's a T7, so T7 is a virus, okay? And a virus, so it's a virus RNA polymerase. Viral RNA polymerases have evolved. Which genes do you think that they have evolved to transcribe if it's a virus polymerase? That's too specific. You're kind of getting close, too specific, but be a little bit more broad. So let me rephrase and say again, or reiterate. The question is, a virus RNA polymerase has probably evolved to transcribe what genes? Or genes from what organism? Okay, so let's think about that. Okay, so that's an interesting answer. The answer that was suggested by a student was that perhaps the genes have evolved to express genes of the host. Think about that. Okay, if you're a virus and you inject, okay, bad drawing of the virus, but you get it, E. coli, you inject your, or you inject your genomic code, it gets translated, and you make your RNA polymerase. Is it going to be helpful for that virus's polymerase to transcribe all the genes of the host? Yes. Okay, so now you've changed and corrected your answer. So instead of actually translating genes from the host, sometimes this thing goes like wacky and doesn't let me like move to the left. Come on. There we go. So instead of transcribing genes from the host, rather, the T7 RNA polymerase has evolved to specifically transcribe its own genes that are T7 genes, so viral genes. So just get the gist that viral RNA polymerases are real good at transcribing viral genes. How does the T7 RNA polymerase transcribe preferentially viral genes? What do you think it recognizes? A polymerase does not recognize RNA, it would recognize sites of DNA. So what sites in the DNA would the viral polymerase recognize? Which is regulated by what? No, not natively. Yeah, the T7 promoter. Okay, so here we have a special gene that has evolved from a virus to recognize a special sequence in the DNA, which is called the T7 promoter. And the T7 promoter, guess what it normally does? Is it normally is controlling expression of the T7 virus genes. So essentially, its own virus polymerase recognizes its own genes preferentially. So that's built into the evolution of this polymerase gene. Okay, so now let's look at the engineered system. I'm gonna go insane if it like keeps like not letting me grab this little thing. Okay, so let's look at the engineered system. And let's walk through what actually happens when we add the inducer. So what's the inducer going to be in this system? We have this plasmid in this strain genotype environment. What's going to be the inducer? Yep. Okay. So we're going to add IPTG. So let's say we add IPTG to the media. Okay. IPTG gets incorporated or sucked up by the cell and it starts turning things on. Okay. First thing that's going to happen is, remember, the LAC system operates by having a repressor. So right now there's a repressor, the LAC-L or LAC inhibitor, sorry, LAC-I. There's a repressor sitting at two sites. Okay, so right now in this cell, there are abundant repressors, LAC inhibitors, holding this gene off and holding this gene off. When you add IPTG, okay, both of these repressors are going to fall off. Okay. So now there is no longer a repressor over here, and there is no longer a repressor over here. But this gene is not actually on yet, okay, because this has a double promoter, which is sort of like two on-off switches. So this one is off currently with this state, but on currently with this state, okay? So this gene is not fully on yet. This one is now, once the repressor falls off, this gene is now completely on, okay? So the next thing that's going to happen is this T7 RNA polymerase gene will get transcribed into messenger RNA, translated into protein, T7 RNA Pol. And this T7 RNA polymerase is then going to be attracted to its own promoter, which has been incorporated into the plasmid, okay? And then as soon as this T7 RNA polymerase goes to here, now this gene is like double on, and now you get expression. Okay, So this is the actual intricate details of the T7 RNA polymerase. It's kind of bizarre. And when you first learn about this, you kind of wonder like, well, I don't understand the complexity here. I don't understand why they would do this double promoter in the plasmid. It's not quite clear or readily why you would do that. So the answer to the reason why you would do that is it's a tighter regulation. Remember in the past, I talked about the lac operator as kind of like a leaky faucet where you even though the repressor is on, sometimes you get a little bit of leaky transcription. If you do this double promoter system, they've figured out a way to make it much tighter. So essentially having these two promoters improves the tightness of the system where it's no longer leaky because you have to sort of have two events that happen that turn on gene X. Okay, so it's essentially just making the system a little bit tighter. And the RNase, the T7 RNA polymerase is a very strong polymerase, and so you'll get very high expression when you do this. So this is kind of the complicated details of the PET system and how it works. Are there any questions about that? Okay, so at least what you should understand on a surface level is that there are these PET plasmids that have this double promoter system, and they are specially compatible only with strains that have an insertion of the T7 RNA polymerase gene. Okay, so that's the gist of the PET system. Now that we understand that, go into the introduction details here. Again, I pulled out things that I think are worth discussing. Okay, so let me look at these. CRISPR-Cas9 system components can be delivered. Okay, so here's what I was discussing before, and that the paper is essentially saying that you can deliver the CRISPR elements in different systems. Here it says you could deliver the plasmid, or you could deliver the system components on a plasmid encoding both the Cas9 and the single guide RNA. So in one situation, you could deliver a plasmid that looked like this, where you had the guide, that's real bad writing, but that's guide RNA, and then this is the Cas9. So here's one way you could deliver and assemble the RNP. The second way is you could deliver Cas9 messenger RNA of Cas9, and messenger RNA of the guide RNA. Okay, that's another way to do it. And then they say there's a third way, which is delivery of, number 3, delivery of the actual Cas9 protein already bound to the guide RNA in the form of the RNP. Okay, and they talk about how there's different pros and cons to each of these. So one of the pros of this system is that it's a single entity. So if you just have this single entity, it makes the reactions a little bit more simple. But what are some of the disadvantages of this system? Of the plasmid double of the plasmid that has two things in it. What's one of the cons of that? Well, the bacteria will always keep a plasmid because there's an antibiotic cassette. But in this case, you'd actually purify from the bacteria and you'd microinject this into like an animal. They did say that. So they did say that one disadvantage of providing this is if you are injecting an organism with nucleic acids, especially DNA, you can get sporadic integrations of that DNA randomly where it can randomly recombine into the genome, and that can cause problems where you might get a mutation. It might insert into the genome and cause problems. So you could essentially give the organism cancer if you're injecting it with plasmids. I won't go into injecting other forms of nucleic acids, but that's definitely one problem with that. Okay, what's another con? So one con was integrations, potential integrations. What was another con? Did it say that? If that you might get off-target effects from the integrations. I would consider that like a subset of integrations. But there's one other thing that you might have missed. I think it says that like you could get some kind of immune response, although you might get an immune response from this, and you might actually get an immune response from this. So that's not unique to this one. There's something specifically about unique to this one that's a con. So this one is slower. Why is this one slower to actually like do the action? I wouldn't necessarily associate it with rare codons, although definitely if the Cas9 is not codon optimized, that would even make it even slower. But you're getting to a point, which is one of the reasons it's slower, is if you inject a plasmid, there's again 2 steps in the process that have to happen before it can do a genome edit. The first step in the process is these things have to get transcribed. The second process is that Cas9 has to get translated. So that's process 1, process 2, and then maybe even process 3 is this thing has to find this thing and assemble the RNP. So do you see how there's like 3 things that have to happen in order for you to get genome edits by injecting just a plasmid? That makes this thing slower. And so you can get reduced efficiency because you're adding things that need to happen to get the edit. Whereas in this system, if you did injected just messenger RNA, there's pros and cons to this. This one might be slightly faster than strategy 1 because the Cas9 just has to get translated and then they have to find each other. So it's one less step. Transcription does not have to happen. But this one has a different disadvantage. What's the disadvantage of injecting the messenger RNA? Yeah, what you said it right, Chelsea. Not stable. Yeah, so remember working with RNA is RNA is not as stable as DNA, and so you kind of want to avoid working with RNA if you don't have to. So this is not really stable, and so you might actually get less efficient genome editing if you're injecting messenger RNA. Whereas the third system where you're actually injecting pretty much essentially the assembled RNP, essentially no steps have to happen. It's already immediately ready to genome edit, okay? So that's one of the advantages of this system. What's the disadvantage of this system? Yeah, it's hard to make. You actually have to do the recombinant protein expression to produce the Cas9, or perhaps you buy the Cas9. But the disadvantage is that you actually have to put in the work beforehand to produce the molecules, but you might actually have more efficient genome editing. Okay, so they have sort of reiterated the point that I described in a previous lecture, that there's different ways to deliver these components and assemble these components. One of these systems, number 3, implies that you can do recombinant protein expression of Cas9. And so this is a paper then figuring out how to do the recombinant protein expression of Cas9. Okay, this is a paragraph where they actually like talk about all those advantages, disadvantages. Let's see. The method provides better control of the intracellular concentration. Okay, so here's another thing that they're saying that's good about the delivering the RNP directly, is that it provides better control of intracellular concentration of the CRISPR-Cas9 system, more rapid activity, and they say decreased immunogenic responses. That's probably variable depending on the organism. Okay, more stuff. Cas9 is a large protein. We already talked about that. It's large size. It has rare codons. So one criticism of this paper is that most people nowadays probably have or are working with codon optimized Cas9. So this is kind of an odd paper in that it's trying to figure out recombinant protein expression of a non-codon optimized sequence. If it were me, I would not waste my time doing that. I would immediately codon optimize and then optimize strains and media based on expression of the already optimized codon optimized sequence. They did not codon optimize. So this is kind of a unique paper in that they're working with a sequence that's not codon optimized. Nowadays, I think this paper's like, I can't remember. I can't remember what year, but I think it's like at least 5 years old. Nowadays, there isn't really even an excuse to work with non-codon optimized because there's so many plasmids that have Cas9. 2019, so that's 5 years. Yeah. Okay. Now then let's get into the methods. So there's some things that are worth talking about in the methods. So here they're talking about the actual plasmid that they're working with. So here they list out the plasmid that they're working with is PET 28B Cas9 His. That's the name of the plasmid. So you should just immediately be able to interpret what that is. You know it's a plasmid and it's a PET system. That tells you a lot, that it has an IPTG inducible T7 system. 28B is just more a specific subset of this. The 28B tells you that it's kanamycin resistant plasmid, I believe. Yes, it's kanamycin. So the kanamycin selectable marker. The Cas9 is the insertion that's inserted into a multi-cloning site, and then His tag after the Cas9 implies what? But where? At the end, which is called technically what term? You're right. You're absolutely right that it's at the end of the gene. What's that actually called? There's a name, specific name for that. Nope, N is in the beginning. So we should review this. Remember, every time you're stringing together a peptide string, on the left side is the amino terminal that is not attached to anything, and on the right side is the carboxylic acid that is not attached to anything. So this is the N terminus, this is the C terminus. So in this case, they have put the his tag on the C terminus, the right side, the end of the protein. Yeah. Oh, okay. Well, okay. Can anybody help them? Yeah, it's an affinity tag. So the technical term is that it's an affinity tag. His6 tags encode the ability to bind to cobalt and nickel. And so we'll actually talk about that purification strategy. But I point out this plasmid because you guys, again, should have all the knowledge that they actually list Addgene. You can actually click on the link and you can pull up the Addgene and you can actually see the plasmid. And I pulled out the map here just so we could look at it and clarify some things. So let's look at the plasmid. Okay, it's got a Cas9 insert. Here's the Cas9. It's got the lac operator right here. Okay, so it's a little hard to read this because the gene is upside down going this way, but the lac operator is going this way, which is what I said. It's already got like sort of like one inherent promoter. And then here it's got a site that says T7. This is the second T7. That's the second T7 promoter. So it's got the double promoter annotated. What's lac L or lac I, sorry? That's the inhibitor. So it's got the repressor encoded into the plasmid. And then it's got some other things, some other origins. And is it annotating the his tag? I don't see the annotation of the his tag, but it should be essentially like right here. Okay, so you should be able to look at the plasmid, understand what it's doing, understand the parts of it. Okay, the medias. Two different medias were used. They're using LB, which we're familiar with, and they're using a different media called high density fermentation media. So they're comparing LB and HDF to see any differences. Here's where they're talking about their purification scheme. So what they're going to do, this goes to Alex's question, which is what they're going to do is they're going to set up a column. And in that column, they use actually in this protocol, nickel beads. So there's going to be nickel beads and they're going to run their, what's called the lysate over the column. Okay. And they're going to drip off and they're going to, after washing, they're going to collect their elution, which is going to have their Cas9. So that's going to be their purification strategy. Here's where they're telling you like what kind of buffers they're working with. So it says after harvesting, the pellet was resuspended in 20 mils of absorption buffer. Here's the buffer. The cells were lysed in an ice water bath. So they collected the cells, they lysed them in an ice water bath with 8 cycles of sonication. So we talked about that. We should know now how they're lysing it. They have a sonicator. This is just telling you how they run the program. And then the crude extract, supernatant. So the supernatant, that tells you that the Cas9 is soluble, was obtained by centrifugation. So they centrifuge the broken stuff down, take the soup, and then the Cas9 was purified by chromatography. That's what I mean, running over the column. And then they quantify their yield via SDS page and densitometry. So at this point, you should be able to sort of read this paragraph and sort of visualize and understand everything that they're doing, the methodology in this paragraph. Here's where they more specifically describe what Alec was questioning about. Cas9 protein purification was performed by gravity. So they're just letting gravity pull the liquid through the column, containing NTA, that's nickel resin. So these beads are made up of nickel metal. Let's see, they're telling you how much they run over it. What I was looking for. Here's what's interesting that I wanted to point out. It says the absorption buffer containing 500 millimolar imidazole. Why would they have imidazole in their absorption buffer, which is where the condition under which they want the Cas9 to bind to the beads? Does anybody first remember what imidazole is? Yes, well, not with the Cas9, but with the his tag. So imidazole is a competitory chemical that will bind to nickel and compete with binding with the his tag. Why would they have a little bit of imidazole in their adsorption buffer? Another word for this, you could call this the binding buffer. So this is the buffer under which they want Cas9 to bind to the beads. Why would they put a little bit of imidazole in there? No, so here's where this is exactly what I wanted to point out. So typically in I think what you're referring to is what's called elution. If you want Cas9 to get removed from the beads, that's the elution step. In elution buffers, it's typically, I think it's typically, somebody needs to look. Can somebody, does anybody have your computer? You have your computer. Can you quick fact check what is a standard concentration of imidazole in an elution buffer? But in the elution buffer, you have a very high concentration of imidazole. Although this is pretty high. But essentially, okay, what I want to tell you is that sometimes people will put a little bit of elution agent, the imidazole, in their wash buffer because that makes the washes more stringent. So you'll lose a little bit of yield of the Cas9 because a little bit will outcompete and drip off some Cas9. But in general, you'll end up with a cleaner product because your washing is a little bit more stringent. So if you need more stringent washing to wash away impurities, people will add imidazole in their buffer. Did you have any success finding it? I don't like to deviate from the lecture and waste time, but I do kind of actually just want to look quick. What? Okay, so you said standard is 500? Okay, so standard, that's weird. So here's where that, okay, if that's true, then they might have miswritten this, or maybe we've detected a mistake, or maybe this is what they actually mean. But I agree with that. Elution buffers are typically 300 to 500 millimolar imidazole. So you're going to get a washing, or you're going to get a complete stripping of all the Cas9 pretty much under these conditions. Typically, I think if I remember, wash buffers typically have something like 0 to 20 millimolar imidazole. Is that what you said? 0 to 50. That sounds about right. Okay, so just understand. Okay, here's what you need to understand. You need to understand that sometimes the wash buffers have a little bit of the thing that causes the elution to create more stringent washing. And the elution typically has a buffer that's constructed of way more, in this case, 10x more imidazole. Okay. All right, now we're finally looking at the results. Okay, here's figure 1. So figure 1, let's look at A. A is a Western blot with some funky colors. So they obviously have a very nice like digital Western blot reader. Looking at Cas9 expression, okay, so here's where they're telling you actually the size of Cas9, which makes sense because here's a 120 marker and above that would be about 150. So digitally or visually, this makes sense. The Cas9 would be up here. And they list out what each of these lanes are. Okay, so M is a marker. It makes sense. Lane 1 is BL21DE3 Rosetta lysate before induction. So this is before induction total cellular lysate. 2 is 4 hours post-induction. So here's what's interesting. You would expect, if you had real strong expression, you would expect to see a bunch of Cas9 right there. They did not see that, okay? And that's a little bit weird. And then 3 and 4 is when they actually start to purify. So 3 is fraction 2 after the elution. And the next lane is the next fraction after the elution. So what you need to understand is they have a column. Again, and they have a column. They're collecting elutions in various fractions. So like the first, right when they add the elution buffer, the first drips that come off, that's what they're calling the first fraction. Then they take that tube and then they put a different tube, a second tube, and they add more elution buffer, and that's a second fraction, okay? So they're collecting multiple elutions. And so here you have, in this one, you have a little bit less Cas9 in the second elution and more Cas9 in the first elution. So this is essentially purified Cas9 in the Rosetta cells. Then they do the same experiment, exact same experiment, except they're expressing in BL21, okay? What do you immediately notice about the same experiment in BL21? Way more expression. Like this is good expression. This is not good expression. Even in the total lysate, which is right here, like global cellular protein, you're seeing a massive amount of Cas9. So obviously the conclusion is here, which cell line is better to express Cas9? BL21, which is bizarre because the whole hypothesis was that Cas9 has these rare codons and Rosetta should, in theory, help express the rare codons. So did anybody catch the rationale for why they think that the Rosetta cells were not expressing the Cas9 very well? Yeah, they said, so again, the Rosetta cells have this extra plasmid To maintain the extra plasmid, you have to add what chemical that keep it? Chloramphenicol. So you have to add double antibiotics. So they add double antibiotics to maintain both the P rare and the Cas9 plasmid. And they actually hypothesize that they think that that might be why they're getting less efficient expression is when you add antibiotics, you're inherently going to get sort of like a metabolic load that these bacteria are going to have to fight against. And so they think that adding that extra plasmid and that extra chloramphenicol might have actually reduced the amount of expression of the Cas9. So they talk about that. And so it's kind of a bizarre result. But what this suggests to you is that nobody knows how to express any given protein X. You have to empirically do the tests. And sometimes the results that you see might not be logically fit your hypothesis. But you have to become an empiricist, which means you do the experiments, you figure out what works, and you stick with what works, whether you understand it or not. That's empiricism. So they like the BL21 cells. Okay, then B, they run a SCS page gel. What's the purpose of doing this SCS page gel if it's just sort of like a reiteration of the Western blot? Anybody have any thoughts? So they're doing the Western blot, they're comparing the Rosetta again to the BL21. The conclusion is exactly the same. You don't really see much of anything of Cas9 in the Rosetta. You see a ton of expression in the BL21, but why even do this? That's one reason. So one reason is convergence. That's what I call convergence, where you do a different set of experiments and you see if you can see the same result. Yes, that's always a reason to do it. Sort of reproducibility, redundancy, robustness, convergence. What else? Why else do this? So which of these is more sensitive, Western blot or SCS page? Western blot is far more sensitive. Okay, so what they're showing you, essentially, like the reason to do both is you're showing different levels of sensitivity. SCS page is much less sensitive. So if you can see the band in a much less sensitive assay, that really validates that you're getting really good expression. So if you can see the band with a standard SDS page gel, which you can here, that just reiterates that really what you really want to use is the BL21 because you don't need any extra antibody treatments to even see it. It's just very, very robust. The other thing that the Western blot will not give you, because Western blot is antibody based, SDS page will actually show you whether or not you have impurities. So the next question is, does the SDS page show that there are impurities in the BL21 sample? No? So look at the whole gel. Here's Cas9. Are there any impurities in this lane? Yeah, there's one here and there's one here. So these are separate proteins that are not Cas9, that are contaminants of the Cas9 purification. So the SDS page is also very useful in recombinant protein expression to show you the cleanliness of the purified product. This is showing you that in general, it's pretty pure, but there's a contaminant here, there's a contaminant here, and there's a contaminant here, and those contaminants might need to be removed. Okay, then they did this third experiment. What type of experiment is this? C. So on the y-axis, they're measuring quantified messenger RNA, which means it's what type of experiment? QRT PCR. So it's a quantified reverse transcriptase PCR reaction, measuring transcript copies of Cas9. Okay, so in BL21, and then the control you need to look at is minus inducer plus inducer. When they add the inducer, you get a ton of expression. When in the Rosetta cells, compared to the Rosetta cells, when you add the inducer, they're getting almost zero expression. So, or there's no change in the expression. So what's actually the conclusion from that C adds? Yeah, yes. So this actually clarifies that, again, the problem is not the codon usage, because the codon usage would apply a problem at translation. So if the codons were the problem, you would have seen a pattern that looked like this. Okay, but because the pattern is not like that, it tells you it's not the codon bias, it's not the codon optimization or the tRNAs that's a problem. It's a problem actually of transcription, which suggests that maybe you don't have very many cells growing or something like that. So they sort of use this to figure out or to suggest that, well, maybe there's actually a problem of like cellular growth. And then they later go into analysis of cell growth. Okay, we're kind of running out of time here, so I'm going to kind of pick and choose what we need to talk about. Here's where they actually say that. Here's where they're talking about the possibilities of why that might be so. I hated this figure. I always hate figures that have double axes. I think you should never do that in science. Some people choose to do that. I think it's dumb. But what's the point of figure 2? Did anybody actually, was it understandable to anybody? So the first thing to understand is that the top is LB, the bottom is HDF. So they're comparing different medias, top to bottom. So maybe the first conclusion we should ask is, which of these is the better media and why? Why can you conclude that HDF is a better media? Yes. So that's actually the data that you look at that makes you conclude that is if you compare the higher cell concentration of LB versus the HDF comes actually up to a higher cell concentration. Again, this is actually really difficult for me to read because I don't actually understand even which axes you look at, but the cell concentration is higher in the HDF. So they're getting more cells. Okay. Then they sort of like analyze the metabolism. The hypothesis, which they point out here, is that if cells are growing, acetate is produced as a byproduct, and they measure the acetate with these lines that I marked as this like yellowish color. So in the LB, there isn't much production of acetate, which suggests that the cells are not really growing very well. Whereas down here, there is a production of acetate, and here you actually use the left y-axis to compare these. There is a higher production of acetate in the HDF. So they again suggest that cells are growing and producing acetate. And then the glucose measure, what is the glucose concentration measuring? Yes, consumption of the energy within the media. So at least in the HDF, you start with a high concentration of glucose and then it goes down. I have no idea how to interpret this, where like the glucose is starting low and ends low in the LB. Maybe that's because there is no glucose in the LB, and maybe I'm realizing that just now. Yeah, that's true. There's no glucose in the LB. So actually, this might be why that's okay. That's very interesting. We figured that out on the spot. That might be actually the reason that the HDF is a better media. Maybe they're actually like using that extra sugar to grow better. Okay, so that's actually somewhat interpretable, although I hate the double y-axes. I would much prefer to see just different graphs side by side. Okay, let's see, what's this? This is then, okay, so then they figured out, so at this point, they understand the cell line. The cell line that they like is the BL21DE3s. That's the cell line. Then they understand the media. The media that's the best is HDF. So you grow BL21s in the HDF. Then they start looking at the induction temperature. Okay, so these are just standard SDS page gels. Here's a control that shows you where Cas9 is. So just look at this and tell me which one of these temperatures, the induction, so after they induce, they keep it at a certain temperature, which of these inductions is the best? 30, because it has the most robust expression of Cas9. So then you know the best induction temperature is 30 degrees Celsius. Then they take that 30 degrees Celsius and then they analyze how long should we grow them at 30 degrees Celsius. Which of these is the best production time post-induction? 8 hours. 8 hours is the maximum amount that essentially you produce, where if you go even longer, you're actually then getting diminishing returns. You're not actually getting a higher yield. They actually say that 4 hours is sort of the point at which you produce the most Cas9 per, like, I think per unit. But then if you wait, the production goes down a little bit, but the maximum yield is going to be at 8 hours. And you can tell that from this band being the darkest. Okay, so this is actually very, very simple data to interpret, just looking at standard SDS page gels, looking at production of Cas9 protein. So again, you can do a lot, you can analyze a lot by just doing very simple experiments. Let's see, what's this last thing? They did some reactions in a bioreactor where I thought this was actually kind of weird. The idea behind the bioreactor is, okay, once you understand all the conditions, you could scale up production, you could do higher growth cultures in what they call a bioreactor, but 2 liters is actually not that much. When I was recombinant expressing proteins, we grow 8 liters, not in a bioreactor. So it kind of strikes me as weird that they do a bioreactor for, I see this as a small volume. So I think it's a little bit weird. And then this last, not the last figure, we're almost done here, hang with me. This is a SES page post purification. So this is post nickel purification, and they're showing you different fractions. Okay. One thing to point out here is that they're getting, I think this is elution 1, elution 2, elution 3, and elution 4. What do you notice about the different elutions? Gets less and less because the first elution is obviously going to strip off most of the Cas9. So that's readily understandable. Now, when you look at this purification scheme, would you say that this is a clean product or a dirty product? This is very clean, okay? You have very purified, highly abundant product, and there really isn't anything else in that sample. So SES pages sort of always run and shown in recombinant protein expression papers as a demonstrative ability to show how pure the product is. And this is a very visually pure product. So they did a great job here getting the Cas9 highly pure. Okay, this is an important figure. We'll end with this. This is the last figure. What is this actual figure? What type of data is this? It's an activity assay, but what actually type of data is this? What is this? What kind of gel? If you're wondering what kind of gel, what are they analyzing? What type of molecule? Wait, are they analyzing protein? This is actually a plasmid. So what type of gel is this? Yes, so this is an agarose gel. Okay, so understand that this is an agarose gel. This lane contains a plasmid that has GFP. Here's their ladder. Another way you could have easily told that it was an agarose gel is you just quickly look at the ladder and it says base pairs. That means DNA or RNA. That does not mean protein. Okay, so you know it's an agarose gel. Here's a ladder. Plasmids are about at 4,000 kb, so that makes sense. My next question then is, in this lane where they only have this plasmid that has GFP, why is there only one band? When you normally run a plasmid, you should normally see how many bands? You should normally see something that looks like this. You should normally see the concatenated, supercoiled, and the normal. Why do you only see one band? Nope, this has nothing to do with primers. They're running a plasmid. So in this lane is literally just a plasmid. Why do you only see one band? Yes, so it actually says that. It says linearized plasmid with GFP. So that means they took a plasmid that had this GFP gene in it, okay, and they quote unquote linearized it, which means they cleaved it probably by a restriction enzyme in one spot. So they opened it up, and that's why you only see one band. So what they're loading into all these lanes to start as a basic premise, everywhere you see this plus at the GFP, that's a linearized plasmid that includes GFP. Okay, now then look at this lane, which has +4 Cas9, has +4 a guide RNA, and has +4 the plasmid. The guide RNA is targeting the GFP gene on the plasmid, and what do you see happens to the plasmid? What happens? Describe to me what happens to the plasmid. Oh, let's ask a different question. What is this band? No. Okay. Yes, this is just the normal linearized plasmid. So then what is this and what is this? It's essentially no, it's not, none of this is supercoiled. Nothing's getting supercoiled. In order to like randomly get supercoiled, you'd have to add an enzyme that does supercoiling. If it's what's happening to the band, it's getting smaller, which implies what's happening. Yeah, it's getting cut. Okay, it's getting cut by the endonuclease, by the RNP. Okay, so what this is saying is that you can actually visually see the RNP cutting the plasmid. If you have the Cas9 plus the guide RNA and you add the thing that it's going to cut, the plasmid, it should cut it. And that's what you see when you see these things, is you see it's getting cut into smaller pieces. Okay, so here's showing you that what they purified from LB is active and it cuts. Here they do the same experiment with the HDF media, and here's where they assemble the RNP because it has the guide RNA, the Cas9, and you're seeing it gets cut. These other lanes are just other negative controls that don't have a fully assembled RNP, which is why you don't see a cut. Okay, last thing, last question, and then we'll go. Last question is, which of these do you think cuts with higher efficiency? Which media treatment cuts with higher efficiency? LB. It looks like visually, it looks like this one has a much stronger cutting than this one, which is kind of counterintuitive because what they're showing you with the data is that with the HDF, they actually get the highest yield, but it seems like it has the lowest activity. Whereas with the LB, they're getting the lowest yield, but it seems to have a higher activity. So you can just see the complexity here in that if you were trying to decide, well, which of these is actually better, it's a little bit complicated, right? So you can see the complexities and the nuance of recombinant protein expression in this paper. That's it. We don't have much time for much left, but unless there's any questions. Okay, I appreciate you guys staying just a little bit here.